Right, it's a real pleasure for me to be here today, and I'd like to thank the organisers for inviting me. This particular session is going to be on understanding comorbidities and carbon monoxide exposure, which is obviously an extremely important topic. Our first speaker today is Professor Derek Steele, Professor of Cellular Physiology at the University of Leeds. Professor Steele started off doing physiology, so we share some commonality there. I did a physiology degree and a PhD at the University of Glasgow. Took up the lectureship at the University of Leeds in 1996 and currently holds a personal chair in cellular physiology at the School of Biomedical Sciences. So his research has been addressing the molecular and cellular events that underlie excitation-contraction coupling in cardiac muscle, and how these processes are modified by physiological adaptations, disease, or exposure to toxic chemicals. Live cell confocal imaging and electrophysiology underpin much of his work, and current projects include remodeling of local calcium signaling in disease, novel mechanisms underlying cardiac ventricular arrhythmias, and potential drug therapies. So it's my pleasure to invite Professor Steele to talk about the effects of carbon monoxide poisoning on the heart. Thank you.
Okay, so I'd like to thank the organisers for inviting me here today. Now, how do I get to the, is it the next one? There we go. So the group at Leeds has actually been studying carbon monoxide for over 10 years. I only have a short period of time today, so I'm just going to pick out a couple of themes that tell you a little bit about the conclusions we've drawn over a number of years and where we are now intending taking the work with funding from CORT. So we've heard a lot about this already today. So one of the issues, obviously, with carbon monoxide is that you can't tell it's there. It's essentially undetectable without a device. And we've already heard a lot today about the fact that carbon monoxide has a higher affinity for hemoglobin than oxygen, and that has the potential to cause hypoxia. We've also heard in other talks today the fact that there can be other effects that are independent from hypoxia on the target organs. And I'm going to try to persuade you today that may provide opportunities for therapeutic interventions. We've also heard a lot today about how you can be exposed to carbon monoxide, and this is just a very short list. So we hear most about faulty gas appliances. I guess that's never out in the news. And as others have mentioned earlier on today, there's likely to be an increase in exposure to carbon monoxide resulting from the use of solid fuel appliances, given the current expense of heating with gas. So those are kind of routine ways by which you can be exposed to relatively high levels of carbon monoxide. There are also a whole load of other situations where you can be exposed to carbon monoxide. Smokers obviously have higher levels of carbon monoxide in their blood. And they're examples of a group of people who may know that they have comorbidities which predispose them to illness, cardiovascular disease, for example, and yet some of them either don't want to or can't give up smoking. So these are a group of people who may tolerate high levels of carbon monoxide in their blood and other risk factors. You also have situations where people, as a consequence of their work, can't avoid it. Firefighters, for example, people working in close proximity to traffic, pedestrians just walking around can have quite high levels of carbon monoxide depending on the conditions, and those living in towns or city centres. We're actually just beginning to understand a bit about how these chronic levels of exposure can affect health. So one of the things you can glean from the the clinical literature is that if you look back over a period of 10 or 20 years, there are quite a lot of case studies describing effects on the heart. Specifically, clinical reports have shown that carbon monoxide exposure can cause an irregular heart rhythm. So this is just a title from a study a couple of years ago, and a particular feature that's picked out from these studies is typically QT prolongation. So we're talking about the ECG here, and it's a particular part of the ECG that becomes prolonged, and that is associated with arrhythmias. And in this case, they recorded a normal QT interval of around 440 milliseconds, and in the presence of carbon monoxide, this was extended to around 622 milliseconds. Prolongation of a QT interval is a risk factor in terms of arrhythmias, and it's associated with quite a variety of conditions. So how does this all fit together with the ECG, the activity of the heart, and the types of experiments that we've been doing in the lab? So if you look on the left-hand side of this picture, you can see a cartoon showing the spread of electrical activity across the heart. So this is a wave of electrical activity that underlies beating of the heart. And cardiac electrical activity begins, as indicated up in the top left-hand side, in the sinoatrial nodes. This is a piece of spontaneously electrically active tissue. And each beat of the heart begins with electrical activity that starts there and then spreads over the left and right atria in the second figure. And then down at the bottom, you can see that the electrical activity is transmitted down the interventricular septum via the left and right bundles of His. And then when it reaches the apex of the heart, it then spreads up through the walls of the ventricles to induce contraction of the ventricles and ejection of blood from the heart. So this sequence of events happens every single time your heart beats. And just below each of these figures, you can see how this particular part of the cardiac cycle relates to the ECG that you can record clinically from the surface of the body. So just to be clear here, the ECG is a very faint electrical signal. Many of you will have had an ECG taken, which is recorded from the surface of the body. So it is a complicated waveform that results from the three-dimensional propagation of electrical activity through the heart. This shows the ECG across here. We know that it begins with atrial depolarization, shown across here. It's missing in this part here. And then you have the QRS complex, which corresponds to ventricular depolarization. And then there's a gap, and then you have ventricular repolarization over here. So the QT interval is a period from the beginning of the QRS complex to the end of the T wave. And in conditions where you have prolongation of the QT interval, this is effectively the part of the ECG complex that is being extended. If you want to understand what's actually happening in the heart when this is occurring, you really need to go down to the single cell level to understand the underlying mechanisms, whether they're electrophysiological or perhaps other mechanisms that involve intracellular components of the cells. So in our lab, what we routinely do is isolate cardiac myocytes, usually either from rat heart or guinea pig heart. And when you isolate these cells, you can record the electrical activity directly from these individual cells. So this is fundamentally different from the ECG. As I say, the ECG is a very faint signal recorded indirectly from the surface of the body. With electrophysiology, you can actually record the change in membrane potential that occurs across the cell membrane, if you like. And this is a result of the opening and closing, we know, of particular ion channels that produces this characteristic effect. So the cell membrane potential is typically around minus 80 millivolts, And during what's known as the action potential, it rises to a positive level. There's a plateau phase, and then it repolarizes again. And what you find is that interventions that prolong the ECG, you commonly find that they also prolong the duration of the action potential. So if you like, the QT interval, extension of the QT interval can result from prolongation of the action potential in single cells. So you can go away and study the action potential, and you can also study effects in individual ionic currents that underlie the action potential to try and understand what's going on. So how do we actually do it? So this is patch clamp recording. This shows a single rat cardiac myocyte. This here is a glass microelectrode, patch electrode that's brought into contact with the surface of a cell. It's known as patch clamp recording. We've been applying carbon monoxide using two different methods, either by bubbling solutions to produce specific levels of carbon monoxide, or as in other studies that you've seen today, we have been using carbon monoxide releasing compounds. And in the early studies, we started looking at the effects of carbon monoxide on the action potential in isolated rat and guinea pig myocytes. And we then went on, I'm not going to show this information in detail today, we went on to try and understand what's happening in terms of the changes in function of the ion channels that actually underlie the action potential. So this isn't actually in chronological order. This is the second study we did in this on guinea pig cells, but it's particularly nice recording, so I thought I'd run you through this first. So here you have the typical control action potential shown here. And you can see that in the presence of CORM2, this carbon monoxide releasing compound, that you get very pronounced prolongation of the action potential. And we also saw this in rat myocytes. In actual fact, the rat myocyte study was done some years earlier. And this made us very interested because it suggested that there were mechanisms leading to prolongation of the action potential which could explain QT prolongation linked to clinical arrhythmias. Why might you be interested in this? Well, depending on the mechanism that underlies this, it introduces the possibility that a pharmacological treatment, if the property of underlying ion channels is being affected, it might be possible to target the ion channels and correct the effect in action potential prolongation, even although the effect of carbon monoxide, even though carbon monoxide is still present. And that could have therapeutic advantage. It takes some time to wash out carbon monoxide if you've been exposed to it. If it was possible to introduce a drug therapy, if someone was suffering from carbon monoxide-induced arrhythmias, it might be possible to induce to add a drug which could effectively switch off the arrhythmias. Now, I'd just like to bring you down here to this bottom right-hand figure. I mentioned that action potential prolongation is a risk factor for arrhythmias, and we did actually see arrhythmias developing in these single guinea pig myocytes. So this is a shorter time scale. This abbreviated action potential here is actually the control response, albeit in a shorter time scale. And what you can now see is, with more prolonged exposure to a CORM compound, this is 5 minutes prolongation of the action potential. But now, after 8 minutes, you can see that we've got this very odd-looking event happening here. So instead of the action potential successfully repolarizing, it has actually started to repolarize, and then you've got this upward deflection. And this is known as an early after depolarization. It's a well-known type of arrhythmia in the heart. And this typically reflects a failure, for one reason or another, of the cell to repolarize. So, as I say, we'd actually done a comparable study in rat a few years earlier. So this is actually going back to 2012. And in the rat myocytes, we did quite a lot of work, I'm not actually going to show today, to try and understand what the underlying mechanism was. And we came to the conclusion that one of the targets for carbon monoxide was the sodium channel. So the sodium channel is actually responsible for this rapid early depolarization phase here. But in certain circumstances, pathologically, the sodium channel can stay activated for a more prolonged period of time. And that can lead to these odd extended action potentials and failure to repolarize. That's known as the late sodium current when it happens. And we knew from the literature that there was actually a drug which had been developed to specifically target the late sodium current. So we asked collaborators in France who are working on long-term exposure to low levels of carbon monoxide to try some experiments for us where they exposed rats to 500 parts per million carbon monoxide to look in vivo at effects on the heart. I'm just going to explain what this is in order. So sham is basically the control condition, and these are original ECG records from rats showing the effects of carbon monoxide, and then the effects of carbon monoxide in the presence of a drug that inhibits activation of the late sodium current. Now, it's probably easier to see from this graph exactly what's going on. So under baseline conditions, right at the beginning, time zero effectively, you have the control response shown in black, and then you have the initial effect of carbon monoxide exposure. So really nothing happening at all in the very early stages. And you can see that adding carbon monoxide plus this drug that inhibits the late sodium current also doesn't have any effect. But over time, and now we're looking at between 30 and 60 minutes exposure to carbon monoxide, you can see these white bars indicating that the QT interval, which is on the y-axis, is becoming prolonged in these rats. And in other data, we found that this was linked to an increased incidence of arrhythmias in the animals. So this effect actually becomes greater if you add isoprenaline. So isoprenaline activates beta receptors, is the, if you like, the basis of the fight or flight response. It speeds up the heart, increases contractility, increases cardiac output. And you can see that the QT interval becomes even longer in the presence of carbon monoxide exposure. Now, if you look at this final bar here and follow it through, this is carbon monoxide plus ranolazine, which inhibits the late sodium current. You can see that under each condition, if you have ranolazine present, it largely speaking prevents prolongation of a QT interval. So we published this. We thought it was very interesting, and we were hopeful that this provided first clues as to how it might be possible to treat arrhythmias resulting from carbon monoxide exposure. As is so often the case, it's not quite that simple. So in man, we know from the clinical evidence that carbon monoxide causes action potential, QT interval prolongation, and arrhythmia. So there isn't really any doubt about that. And our work in animal heart cells has identified the sodium current. So our early work in rat identified the sodium current as one of the targets for carbon monoxide. So if you go over to the right here, this shows the action potential, and you can see where various currents activate, shown in green here, during the action potential to sort of induce the various phases of the action potential. And you can see that the sodium current activates very early on here, and in the presence of carbon monoxide, you get a very prolonged activation that helps to extend it. And this is present in rat. So ranolazine is capable of preventing this prolonged activation of the sodium current, and that explains why ranolazine so effectively switched off arrhythmias in rat myocytes. The problem is, we then went back to try it in guinea pig, and in guinea pig, ranolazine did not work very well, and we know from further work why it didn't work very well. It's because rats lack another channel, the hERG potassium channel, which is important in repolarization. So this is IKR down here. And we found that carbon monoxide actually inhibits hERG. And the effect of inhibiting hERG is to prolong the action potential and promote arrhythmias. So in a more complicated system that's actually closer to man, man also has both the sodium channel and HERG, ranolazine doesn't work very well. And in essence, what it comes down to is, we've looked at two species. We looked at rat and guinea pig. Rat's a somewhat simplified system where the sodium channel is present but HERG is not, and we demonstrate there that yes, you have the potential to switch off these arrhythmias pharmacologically, even although carbon monoxide is still present. And I think that's a very important point to take forward. But the complicating feature is that it also affects HERG, and HERG is present in man. So you're left with a question, to what extent do these experiments relate to man? And to know that, you really need to have a very good model of human ventricular cardiac cells to try and work out which of these two effects is dominant and which drugs are going to be most appropriate to use, most effective to inhibit arrhythmias. Just to finish off, this explains where we're going now. So in a project funded by COT, we're actually going to be looking at human-induced pluripotent stem cell cardiac myocytes. These are being used increasingly in toxicology studies to overcome a particular problem. Sorry about that. A particular problem, and that is that you cannot easily get cells from human hearts. So you can occasionally get bits of human atrial tissue, very difficult to isolate functional cells from them. It's essentially impossible to get functional human ventricular cells. So drugs companies and the research field, related research fields, have been working in this, and what they've developed is models of the human heart based on stem cells, which it turns out are very good at predicting effects on ion channels, including the sodium channel and HERG. So the study that we're going forward to do, funded by CORT, will be using this model to try and understand how to target these arrhythmias effectively in a system that we hope should have most close relevance to man. I just want to finish off by acknowledging some of the people that have been involved in this work over quite an extended period. So these are the group of people that have been involved at Leeds. And I must also mention our collaborators in France who did the in vivo work on rats. The work has been funded over the years by a number of people, mostly the British Heart Foundation, also Gilead Sciences, and more recently the Carbon Monoxide Research Trust. Thank you.
Thank you, Derek. That was a nice reminder for me of why I don't like physiology, because when you think you've got the answer and you try it on another species, it all goes out the window. Do we have a very quick question? Oh, okay. Thank you again, Derek. Right, next speaker is Mary Herigstad, who I've only known virtually for the last 2 years, which reminds me, you're on mute. Mary did a DPhil at the University of Oxford, where she studied cardiorespiratory physiology, and following a postdoc work on neurocognitive patterns in patients with smoking-related diseases, she became interested in the effect of carbon monoxide on physiology. The current focus of her research group is on how low-level exposures affect vascular and neurocognitive function, embryonic development, and select cellular pathways, using a combination of human physiological and imaging research, model organisms, and cell biology approaches. And the work that Mary will be talking about today is funded by the Carbon Monoxide Research Trust.
Thank you, Rob. Thank you for reminding me of 2 years of cursing at my Wi-Fi. Yeah, so it's nice to see you in person. That's really great. So let's see if we can get this to work. Marvelous. I'll be talking about the effects of carbon monoxide on the brain today. It's going to be a little bit of a combination of some old results and some new ones, so I hope it'll be interesting. The level of carbon monoxide that we are using, since we are dealing with human beings, is very low. So I thought I'd start with that, because obviously we've been discussing what's low and what's very low and what's high, and there is no consensus, so I thought I'd just show you. We have been increasing exhaled carbon monoxide in our volunteers from around 3 parts per million to about 6 parts per million. We chose 6 parts per million because it's been associated with smoking. It was the lowest level. That was one of the reasons we did this work in the first place, to see if smoking was a confound for fMRI studies. So that's why we chose that level. If anyone wants to know the carboxyhemoglobin level for that, I think it sort of corresponds to 1.1% up to 1.5%, so it's low. We did check, by the way, that the exhale level and the carboxyhemoglobin corresponded in me. We poisoned me a number of times, and it corresponded. We did the assessments at the same time. So we're pretty confident that that is this level that we're talking about. What we're looking at to go forward, just to give you a flavour of what we're planning on doing, is to look even lower than that. So we're not interested in higher levels. We're not interested in poisonings, direct poisonings. We're interested in exposure that we'll get from out on the street, etc., etc. So that's the level we're talking about, up to 6 parts per million. And that's the sort of thing you can get ethics to do in human beings, which is great for our work. We've been using an imaging technique called fMRI, and I know some of you will know a bit about this and some of you will not, so I thought I'd explain very quickly what we're talking about. fMRI, or blood oxygen level dependent fMRI, which is what we've been using, is an indirect measure of neurological activation. So what happens, say for example, you got your brain, there's blood going to it all the time, but if you use a region a bit more, then the theory is that there will be an influx of blood to that part of the brain. So, for example, you look at a flashing checkerboard, there will be an influx of blood to the visual center at the back of your head here. That's the theory. So that's the little graph that you see there. And that influx of oxygenated blood will be greater than the requirements of the region, typically. It'll also be a little bit delayed. Now, the magnetic properties of oxygenated blood is different from deoxygenated blood, and it's that we're picking up on. It's the key is in functional magnetic resonance imaging. So you're picking up on an influx of blood with a certain magnetic property. Now, the fact that it's indirect is relevant for the rest of this talk. What we're also doing with fMRI is that we're looking at a baseline, so not looking at a flasher checkerboard, for example, versus looking at it. So you're always looking at something in relation to a baseline, because as we're sitting here, there's blood going to our brain all the time. It's not like there's no blood and then some blood. It's always blood and then there might be more blood. So that's what we're looking at. And in terms of explaining the figures, they are usually pictures of, colors on a brain, and increased activation will be in the area of red to yellow, with yellow being greater level of significant activation, and decreased activation, which we can also look at, where blood flow might go down with the task, is in blue to light blue, with light blue being the bigger decrease. So that's just an overview. I hope that made sense. We got a couple of tasks that are typical for fMRI that we use as a way of assessing how the neural function matches the blood flow, or just in general how the blood flow to the brain is in response to a stimulus. These are shown here. The top one is a visual activation task, which I've already talked to you about. It's a flashing checkerboard, and it generates this very robust signal at the back of the brain, and we use it to assess the coupling between neural function and vascular influx of blood, because most people have this. There are exceptions, but we don't scan those. We know that this is predictable, and we can tell then if someone doesn't have the predictable blood flow in this region, that the neurovascular coupling is off. The bottom one is a breath hold, and that's interesting because you get a massive global increase in blood flow. As you can see here, it's pretty much the entire grey matter of the brain that's lighting up, and that's because as you hold your breath, carbon dioxide rises, and carbon dioxide is a vasodilator. So from not holding your breath to holding your breath, massive influx of blood, and that's what that picks up. And again, that shows you how well your vasculature in your brain responds to a stimulus, in this case, a breath hold. So the things that I'm going to talk about are 4 areas. The first one is visual processing with carbon monoxide or without, and the other one is the breathing, breath hold. So that will be reflecting those 2 examples there. We're also going to talk a little bit about cerebrovascular function and end up with some behavioral cognition data that we just got. So in the interest of keeping to time, we're just going to get started. So this is the visual processing data that we've got in healthy volunteers, 12 in total, that we exposed to that level of CO that I talked to you about on the first slide. Flashing checkerboard versus not looking at one, on and off, and then we look at the baseline versus watching this checkerboard. And if you look at the picture all the way towards the end there, that's mean activation. The top one is before the person inhaled CO, and the bottom one is after. It doesn't really look like there's much difference, but you can see that there is this nice visual activation. If we look at the contrast between before and after, and we compare that to an air control protocol, because you need to compare to air control, otherwise you don't really know if it's CO or if it's just the order of which you're doing the scans. If you look at that, you'll see that there is a reduced activation in the visual cortex for this task. Now we can quantify this. By the way, we didn't see this in the air protocol at all. We did not see this reduced activation. Now we can quantify this by looking at individual percentage change in the Fmri signal and match it to the individual exhaled carbon monoxide levels. And we see that the greater reduction with, in signal is associated with the higher levels of carbon monoxide inhalation. We can also see, so the B figure there is the area in which that activation is reduced. So that means, according to that data, we see either a difference in neurovascular coupling or we see a reduction in activation. As has been mentioned before, it's never always that simple. So it's going to be this is going to be a talk of caveats. So, but we do see an effect of carbon monoxide, this low level of carbon monoxide. So we can look at the breath hold then, and the top one here, the top graph shows the breath hold, just a rise in CO2 as the person were doing the task. And again, at the far end there is the mean activation. That's the top one is before CO inhalation, and you can see it's a massive global response. The bottom one is after CO inhalation, and that's not a mistake, that's a complete nothing. Nothing reached significance. It doesn't mean that there's no activation, but it means that there was nothing that was significant with the task. So I was a little bit flabbergasted when I saw that. We can look at the same thing. So the top one here on the closer figure, that's A, that shows the areas in the brain that are significantly reduced in activation with the CO. There are less there than you'd expect, judging by the mean data, but that's because, as I said, there is activation in the bottom picture on the far end. It just doesn't reach significance. So you are deducting something, so the comparison is always going to be less than what mean would suggest. The interesting thing is that we are seeing this reduction in activation not globally, but in areas associated with breathing, things that we are used to seeing. Brainstem, so cardiorespiratory areas in the brainstem, areas in the motor cortex, areas in the insula. Figure B there, shows the same thing that the graph showed in the previous slide, just on a brain, showing the area of the brain where the reduction in activation correlates with the amount of CO that the person is exhaling. So that's individual levels compared to individual levels for each person. And again, I've got another figure here, and that's number C, and there's different data from smokers and ex-smokers, an older group, and I wanted to include it because I did this analysis and I found delayed Fmri activation in this very same region in the ex-smokers, but not in the never smokers. This suggests that there might be some lingering effects. We have no idea if that is the case, but it's something that I really like to look further into. Now, as I mentioned, these are standard tests. They assess neurovascular coupling or cerebrovascular reactivity. So that's, it could be an effect of neurological function, or it could be an effect of vascular changes. And carbon monoxide is a vasodilator, so it is possible that this is just vascular. There are a few things that indicates that it may not be only vascular, and that is that the regions that we see are reduced in activation are very task-specific. They're related to the task that we're asking the person to do, and if it hadn't been neurological in nature, we might expect to see it more globally. And the other one is that lingering effect in the ex-smokers that I talked about. So that suggests that they might be neurological as well. But in order to really figure out what's going on here, we've done a couple of subsequent steps. And one is to determine the global vascular reactivity to see, is there a change in vascular reactivity? The other one is to do a behavioral test to see, is there a behavior in neurological function? These are pretty logical next steps. So this is the cerebrovascular reactivity and neurovascular coupling analysis that we did. So what we did is that we took the signal for the breath hold and we converted that, we could convert that to percentage Fmri signal per millimeter mercury of carbon dioxide. That means that we could calculate how much signal would go up with each millimeter of mercury of carbon dioxide during the breath hold. So say, for example, a normal air-breathing person, you could say you have 1% of signal with each millimeter of mercury of carbon dioxide. And if that was reduced with CO, you might just see a 0.5% increase in signal with the same amount of carbon dioxide. I hope that makes sense. So just to make a long story short, we did see a reduction in cerebrovascular reactivity. The picture that you see there is cerebrovascular reactivity in air, nice bright yellow, big signal, and the one next to it is slightly redder, that is with CO. That suggests that there is certainly a vascular component to our findings. And the reason for this might be as simple as this. So here is a suggestion of what happens in the blood vessels with air and with CO. The first one is air. You've got a blood vessel that is, nice and relaxed, and then you hold your breath and you get an influx of a vasodilator and it expands. Marvelous. It's dilated, you get a big signal because there's a big influx in blood. If you have a pre-dilated brain because you got an increase in a baseline because of your inhaling CO, then it might only dilate a little bit further. There's only so much that the vessels can dilate. Therefore, you'll get a reduced perceived activation. You get a reduced signal. It just can't dilate any further. So that might be the reason for that. Now, we've also done a test to look at a behavioral task. And the reason for doing that is that if we do a behavioral task, we can scale activation to what people are actually doing with their performance, giving us a better idea that this is neurological in nature. So this is an analysis that's been done by my PhD student, Lucy, who's there today, her first conference. And it's work in progress. It's quite new. We haven't even gotten close to publishing this yet. Hence, it's not the most polished figure, so I apologize for that. But what we're seeing at the far end there is post-air at the top and post-CO at the bottom. And it looks quite similar. But if you look at A, which is the contrast again, before and after CO compared to air, there is areas that are reduced activation after CO, again reduced. Apologies for this not being in blue, light blue. Again, as I said, it's hot off the press. So what we're seeing is this is an area again, it's task related, audio spatial awareness in the temporal lobe. It's kind of what we'd expect with a task like this. And if you look at the behavioral data next to it, there isn't any significant difference between protocols, which suggests that the reason we're seeing a difference is not because after CO people just decided, no, I'm not going to do it, I'm just not going to respond. The response is more or less the same, not significantly different, but there is a difference in how the brain responds. So we thought that was really, really interesting. So just to summarize, our data suggests that there is, at least for this low level of CO, acutely administered, there is a neurological component and a vascular component, so it affects both. There are a number of things that we don't know, though, so again, things aren't simple. We have no idea how much is vascular and how much is neurological. We don't know that yet. We don't know the underlying mechanisms. We are currently starting some research funded by Court to look at potential target molecules, nitric oxide, that sort of thing, and try to figure out what exactly is the mechanism behind the vascular side of things and later on the neurological side of things. Another thing is that Fmri is very expensive, so we only could do this in a few number of people. What we want to do is to see if this is more general, so we're looking to use a much larger sample. There is the UK Biobank, and they've got MRI images from 25,000 people, so we are going to get access to that and try to pair that with atmospheric CO levels, and hopefully that'll either verify or at least give us some more information about how this works on a greater population level. Another thing we don't really know is dose dependency. So this was an increase in 3 parts per million for these people from 3 to 6. We don't know when that effect kicks in. We're wanting to look at much, much lower levels and get a dose dependency curve. Now, that's not easy to do with fMRI, so we decided to use some other imaging modalities, transcranial Doppler ultrasound, which will give us a beat-by-beat measurement of blood flow in the brain. So that's something that we are starting on doing now. And we're also wanting to explore how this links with disease longer term, because impaired cerebrovascular reactivity, as we've seen, whilst it might be just a side effect and it's interesting for fMRI studies, it might also, if it's chronic and low level, it might affect a risk of disease longer term. So we're looking at this data here, which is the sort of environmental data that's from Leeds, London, and Edinburgh in 2019. And the levels that we see there are associated with epidemiological studies showing increased disease risk, vascular dementia, heart failure, stroke, and so on. So we'd really like to see how our findings match with these studies and look at mechanisms and see whether or not prolonged exposure is indeed dangerous. So that's the sort of thing that we're currently looking at, and kindly sponsored by Court to start digging into in some detail. And only the last thing for me to do is then to just thank the people that have been involved, Lucy, and also my colleagues at SHU, Prachi and Caroline. Prachi's here today, and co-workers at the University of Sheffield. And this work has been in part sponsored by Court and in part sponsored by the Oxford Brookes University, so it goes a couple of years back, at least for me. And with that, I think that's me done. Was it within time?
Thank you, Mary. Ooh, I'm on mute again. Thank you, Mary. For those of us who've got no idea what we're looking at when we see a picture of a brain, I would have thought that the changes would have been symmetrical, but quite a lot of those, the changes were just on one side of the brain.
Yes, so it's not always symmetrical, and there are a lot of studies that have looked into this. There are many types of tasks that will be bilateral, but some are not. Anything to do with reading will often show preference to one hemisphere to the other. The behavioral data that you saw that was very much to one side might be simply because they are responding with a button box with one hand and it's on the opposite side of the brain. So some things will show up like that. The interesting thing about at least that analysis is that you see the activation on both sides, but it's more pronounced and only reaches significance on one side. Again, it would be nice if we could have some more volunteers, and we might see it turning up bilateral as well. But yeah, that's pretty common.
Do we have any further questions for Marie? Shirley?
Oh yeah, you can shout.
We know you can shout.
So Marie, this is really a comment more than anything else. You're going to look at the biobank work, which I think will be fantastic. So are the questions that you may ask, looking at what you've got here, is considering things like age, disease state, and the continuation and perhaps looking at adaptation and reversibility?
So that's a really good comment, and yes, we are hoping to incorporate some of these, not all of them. Carbon monoxide exposure we'll have to do that with data from NASA. They do sort of a CO grid of the entire world with surprising accuracy, I think. So that's what we're using for that. In terms of age and demographic details, the biobank will have a lot of those. And we hope to match it in so that we get a good model. It's a big, big task. It's 25,000 people, so give me some time to get through it. But definitely some of the things that have been mentioned today have been really interesting in terms of writing down, we'll need to account for that, we'll need to account for that, we'll need to account for that. The more we can put into that model, the better it will be.
And I think And I think that's where we're looking at. It's actually looking at models and modelling this. So even with 25,000, you can look at a random selection. And also gender would be very important to consider, I think, as well. So there you are, another 10 years worth of work.
Yeah, I know. And also it would be, as you say, with modelling, I think that might be one of the things that we are looking at going into, not only doing the analysis, but seeing if we can match something with mathematical modelling of biological function, because that would be really strong results for this sort of thing.
Izzy.
Thank you, Maury, for a great presentation. This is not really a question, it's more of a plea. Some of the SEER Research Trust members might not recall that as GST, they funded some aspects of Biobank and put money in so that they would capture people who had gas appliances. So when you look at your Biobank cohort, that's another factor that you might like to take into consideration.
That is actually news to me. I didn't know that we did that. But yeah, well done whoever decided to fund that, because it's going to come in handy. I think in general, might be just a personal plug, but I think the more we can have a shared pool of data, a biobank of sorts, the better our work will be as a whole. We can help ourselves and support ourselves. So that previous work will feed into what we're doing, and we hope that some of what we are doing will help feed into other people's work as well.
Do we have any more questions? Okay, thank you, Mary.
Thank you.
Now, our next speaker was going to be Beth Cheshire, but unfortunately she can't be here this afternoon for various reasons. Professor Carol Holland has very kindly agreed at the last minute to jump in to give Beth's presentation, which is fantastic. Thank you again for doing this. So we've heard about effects of carbon monoxide on different organs this morning, and then on the brain and the heart this afternoon. Of course, a lot of people, if you look at the general population, we're quite top-heavy when it comes to the older generation. And I'm putting my EDI training into good use here, Shirley. Thank you. We do know that there may well be a susceptibility of the more aged population. So it's going to be very nice to hear about the effects of carbon monoxide poisoning now on older people. Thank you.
Thank you. Thanks. So I have stepped in rather last minute, and I'm giving somebody else's talk, so do bear with me if it's not quite as fluent as it would be normally. Okay, so Beth is a PhD student. She's just finished her PhD, so she just handed her PhD in 10 days ago, and isn't really very well today, so I've stepped in. But a huge amount of work has gone into this, as you'll see. I'm only going to talk about some of the results, not everything today, but I'm hoping she'll be recovered enough to join you tomorrow, and you can quiz her a little bit more about her studies tomorrow. Okay, so we are looking at low-level chronic exposure, which I think is what a lot of people have been talking about today. There were, as I think people have mentioned, a series of studies in the 1970s, 80s, 90s, which weren't particularly amazing studies, but they were done under very different cultural sort of situations. But they looked at exposure studies of around 5% and found that this kind of exposure was associated with various kinds of impaired cognitive function. And one of the first things Beth did was to do quite a sort of literature review, a very systematic literature review of some of those studies to pick out what are the actual cognitive functions there, and that really fed into the rest of her studies. So we know that chronic low-level carbon monoxide exposure over about 24 hours in terms of duration can result in things like headache and nausea, as we've seen, affective disorders, so depression and anxiety and things like that, but also particularly memory impairments and motor slowing. We've also got epidemiological studies that have shown associations between air pollution and increased risk of stroke, myocardial infarction and heart failure. But also of interest in my area of research is the carbon monoxide exposure being linked to increased risk of development of different kinds of dementias. So high-risk groups, so that's what we're talking about here in older people as a high-risk group. So poisoning severity depends on human and environmental factors, duration, concentration, and of course pre-existing disease. And we all know as we get older we are more likely to have chronic illnesses. So older adults are at higher risk of unintentional carbon monoxide poisoning. Firstly, they may be more susceptible to the effects of carbon monoxide, but also they may be at high risk because they spend more time in the home. But of course, a lot of us have been doing that anyway over the last couple of years, so it's relevant for all of us. But in normal circumstances, retired people do spend more time in the home. So within UK homes, carbon monoxide levels have been reported to exceed WHO guidelines at a certain percentage. So the sort of Croxford studies, 2005-ish, found that 19% of homes had CO levels exceeding the 8-hour guideline of 9 parts per million. And that's frequently associated with gas appliances, and some of Beth's work is starting to show that it's more associated with gas cookers than with other boilers and things which seem to be more regularly serviced. And it's a particular concern in the UK because we use a lot of gas appliances for heating and cooking. So a percentage of the population may be at risk from low-level carbon monoxide exposure. And of course, people are unaware that that's going on, as we've heard in various examples this morning particularly. So the aims of the study were to look at older residents, particularly in the Coventry area. And one of the reasons for this is that the fire officers had basically come to the researchers with this question about why are so many of the older adults we visit showing signs of confusion when they don't necessarily have a diagnosis of dementia. And obviously there's lots of reasons for that, but that's kind of where the conversation started, and the fire service are here today to smile nicely and support Beth as well. So the aims were to examine the proportion of older adults of homes in Coventry with low-level carbon monoxide, and then to examine the effects of that chronic low-level exposures on cognitive function particularly. So the aim is specifically looking at cognitive function. We have got some health measures as well, but I won't be talking about that today. So a sample of 106 older adults, mean age 75 and a half, around about 75 and a half, and they were recruited to take part in quite a long-term study. So we monitored their homes for a month with data loggers that took regular samples of the carbon monoxide in the air and did quite an in-depth neuropsychological assessment. And then we followed them up at 7 months later, so with another month's worth of logging of the carbon monoxide data. So we could examine both the immediate impact on cognition, and we could also examine the longer term impact, but we could also replicate the immediate cross-sectional impact as well 7 months later. So the cross-sectional results first. So that's the impact of the 4 week logging, data logging, the 4-week exposure that we measured with neuropsychological assessments taken at the same time. And what we found was these are very low-level exposures that we're talking about here. And what we found that generally exposure to low-level carbon monoxide was associated with positive cognitive effects. So the opposite to what you might expect, but actually people were the higher the percentage at different levels of carbon monoxide, then the better people were doing on the cognitive function. So this is short term. Okay, and that kind of, if you think about some of the things that we've been hearing about this morning, the effects of endogenous carbon monoxide, that kind of works, especially with older people, which we'll talk about a little bit more in a minute. And these are, You can see listed the levels at which we found these effects and the different kind of cognitive functions that we were looking at. The cognitive functions we chose to look at were based on the earlier literature review. So auditory working memory, memory recognition, visual working memory, visuospatial ability, and so on. You can read them as well as I can. So the the CO levels were extremely low. They were all below about 29 parts per million, with some really very low means, as you can see. So the cross-sectional work was showing an improvement in cognitive function, only very small effects, but really quite consistent. And we heard this morning about the roles of endogenous or internal CO production. It's involved in various cellular functions, vasodilation and proliferation, crucial role in cellular maintenance, protection, regeneration, survival, and so on. And particular cytoprotective properties at low concentrations, to the extent that it is being used for, explored for therapeutic use, as you've heard. And these physiological processes potentially minimise the risk to the CNS at these levels and perhaps play a beneficial role, but we're expecting that's only obviously up to a certain dose and duration, because we know from all the other data that carbon monoxide, it acts as a poison at higher levels. So endogenous carbon monoxide plays a role in vascular tone, and these vasoactive properties can result from low-level inhaled carbon monoxide, as you've seen in some of the other studies, especially the one we were just listening to just now. But aging, as you know, is associated with various vascular changes. So we know that older blood vessels don't expand and contract quite the same way as younger blood vessels do, for example. So what we're suggesting is that this might be a particular effect in older people because they kind of simply have more constricted blood vessels, so it may be a more positive effect for the older people. So older people have various cardiovascular risk factors, commonly, not everybody, but commonly. Things like coronary heart disease is more common as you get older. Atrial fibrillation, as we've heard, is more common in older adults. And that generally leads to a greater reduction in cerebral blood flow and chronic hypoperfusion, so basically not enough blood getting around the brain. And that further compromises the already reduced cerebral blood flow that's present in aging. So the joint effect of age and disease, so you've got age on the one hand having these effects on vascular health, and you've also got potentially increased risk of cardiovascular disorders. So age and disease combined can have an impact in reduced oxygenation of the brain, so neuronal energy crisis, neuronal dysfunction and death, and also increasing the risk of cognitive decline and dementia. And this process is particularly initiated in the sort of ischemic sensitive zone, so the zones very sensitive to a lack of oxygen. So the areas like that we've been hearing about, the hippocampus associated with memory, basal ganglia, and also cortical white matter, so the connective tissue of the brain. So the protective effects of low-level carbon monoxide might be of particular benefit to older adults, and obviously we'd need to do these studies in younger adults to see if we had the same effects, but we would hypothesize that they don't have the same effect to the same extent in younger adults, and some of the older literature suggests that's the case, that these positive effects weren't reported, but that might be reporting bias as well. But the cognitive functions that we see that are improved by these very low levels in the cross-sectional manner are similar to the pattern of performance improvements observed, but also performance deficits we see in more serious poisonings. Okay, so that this is sort of the end of the first study that Beth was doing, and we hypothesized that these positive effects were likely to be transient, and that the areas that are most susceptible to these temporary increases were also the areas that may be most susceptible to damage as well, potentially resulting in deficits in similar cognitive areas to the beneficial effects that we observed in the immediate cross-sectional data. So we then looked at 7 months later, we looked at the same people. We only managed to get 78% of them to follow up at 7 months later, some due to COVID, some due to their age and poor health. And 1 or 2 of them have passed away. So we had short-term effects from time to exposure, so we could replicate the analyses that we'd done in terms of a month of exposure and analysis of this at the same time. But what we could also look at was the longer-term impact. So we could look at the impact of the carbon monoxide levels at time 1, 7 months ago, on what the cognitive function was now. So this is as near to a sort of chronic study as we could get within the scope of a 3-year PhD. So what we found was that cognitive performance at the longer term exposures did start to decrease. So although we could replicate the increase, as you can see in the top half of the slide, increase in performance, we found decreased performance at the longer term, the chronic 7 months level. So we found these in areas like processing speed, intra-individual variability, so that's how variable people are in a reaction time study, which we know is a reasonable a reasonable measure of brain health variability in these kind of measures, but also selective attention, resistance to distractor interference in different kinds of attention studies. So we also found that cognitive performance decreased with greater CO between the following ranges. So you can see these are still all really quite low levels. This is percentage of readings in those particular ranges of carbon monoxide. And again, you can see some of the cognitive functions that are showing a decrease in this slightly longer term analysis. So you can see memory recognition, auditory working memory, and so on. You can see the different kinds of cognitive functions that we're looking at. And the only one that actually carried on increasing was visual working memory at the very low levels. We then looked at the overall exposure. So although we don't actually know what happened in between our month 1 and our month 7, we didn't log the data in between because we only had loggers that would last a month in terms of the batteries at the time. So hopefully, as time goes on, we'll be able to get longer periods of this kind of level of logging. But we had, in terms of the overall exposure over those 2 months that we were able to assess, we still found some positive carbon monoxide related effects across some functions in the short term, but we found these longer term negative impacts as well. So 2 different ways of analysing the data, different ways of measuring the long term exposure effects. So what we're proposing is that these kind of chronic, the impacts of low-level exposure can be viewed on a continuum, if you like. So at the one end, at extremely low-level, short durations, we see potential beneficial effects. And then at the opposite end of the spectrum, we see negative impacts with increasing exposure duration and concentration. And at some point in the middle, there's no effect for some of these variables. If you can see, it goes from negative through 0 effect all the way up to positive 0 all the way up to negative effects at the higher durations and higher concentrations. And this shift, so we tested this by looking at whether there were shifts in these particular variables, and we found this shift in these variables here, so selective attention and resistance to distractor interference, memory recognition, and auditory working memory, and processing speed. So what we're saying is that some of these variables, some of these specifics of cognitive function are more sensitive than others, and it really takes a lot more exposure for some of the variables. Some of them are more resistant before they start showing a negative impact. So when we say that there's varying studies, some studies show that there's negative impacts, some studies show that they're not. What we need to start doing is to look at the specific cognitive functions that we're looking at, because what we're showing is that they're different, they respond differently. Some are more vulnerable than others. So these are sort of domain-specific shifts from positive to negative effects, and that supports this view of a continuum. Some functions were associated with positive effects only, and we are assuming, although we don't know yet, that is because we didn't actually get to the duration or the levels of carbon monoxide exposure at which there may be negative effects. And other cognitive areas didn't necessarily follow the same trajectory, so psychomotor speed, inter-individual variability, and they may be related to negative effects only, given sufficient exposure time, sufficient post-exposure and significant accumulation. Okay, so this proposed model of a CO exposure trajectory kind of increases our understanding of less severe exposures. As I say, it's a possible explanation for some of the inconsistent findings we've got in the literature, and it can account for some of the absence of effects, some of the negative effects, and some of the trends towards positive effects we've seen in the literature. So this careful work that Beth's done in terms of separating out may provide some of the answers to some of those inconsistencies that we've had. And it kind of bridges that knowledge gap between the potential beneficial effects and the toxicity that we're seeing. So in terms of the sort of clinical implications, this might be important to enable us to highlight the specific domains of cognitive function that are affected and may start telling us a little bit about thresholds of harm that we may be able to see. So, as we've said, particular functions seem to be more sensitive to carbon monoxide exposure, and other functions seem to be more resilient to carbon monoxide exposure, with only positive effects observed, and some areas have only negative effects. So the identification of these areas, those most and those least affected, may be adding to our growing armoury of potential markers for clinicians to be able to determine effects on people. Okay, so just to sum up, the sort of vascular alterations observed in ageing and cardiovascular disease may have an impact on both cognitive decline and are associated with dementia development, even outside of carbon monoxide research. But actually what we're suggesting is that some of this might also be adding to the risk of the development of cognitive impairment and dementia. So future research is looking, we're hoping to look at some longer term exposures to go back to those people, and also to start looking at a wider range of exposures. So we've talked about solid fuels and different areas. So this is just based in Coventry, which is a particular kind of housing and so on. So hopefully we can extend this kind of research into the future. I'm guessing I've run out of time.
Thank you, Karen. We've got just a couple of minutes for any questions. While we're waiting for the microphone, I'll just quickly interject. So the population that you studied, you had the monitoring equipment, and it determined that they were regularly exposed to carbon monoxide. When you went back 7 months later, had there been any interventions to reduce the dose, or was it still the same situation?
So if it was over a certain level, then the fire service went in and did an intervention. When they went back to plant the data loggers again at 7 months again, they gave a lot of information out and talked to them about, the guys who did it are here, so you can probably quiz them a little bit more on what they actually did. So they did a sort of intervention at that point as well. So, but if somebody was showing more than 20 parts per million in the house before we started, they didn't take part in the study, and they raised an incident and sorted them out, basically.
I saw a hand up a moment ago. Here we go.
Hi, Hilary Waring from Improving Performance in Practice. I think it's a question for Rob. Is there any and you mentioned it, Shelley, you mentioned it before, about a little bit about gender. Are there any differences in the impact of carbon monoxide in gender or the treatment for carbon monoxide by gender?
I'll pass on that one, thank you.
Henry, I don't know the answer to that. I think it's something that you need to consider in as much as, because of systemic effects, hormonal effects, etc. I don't think we have an answer. You know, all the animal studies, I'd be interested to know, are we looking at only male rodents? Can you get, I'll project. So we don't know. And I would just be interested, you know, the studies that I've done with animal work of many drugs and other chemicals, you always see a difference in the gender, and it's sometimes related to the hormonal status of the animals, and you can start to pick up on various things. I don't know the answer to your question, but I think it's important. But I also think it's important, so it's what's going to lead on with that, is the differentiation of this group of people in things like drugs, smoking, as well as disease states, etc.
It's just that particularly in preparing the paper we've just been preparing with Beth, I just see no mention of gender. And I'm just quite surprised, given that it's talked about a lot in other areas of public health and, generally in other areas of health.
Can I just come in on that one? So in the data that Beth was looking at, she had various measures and various demographics of the population that she had. So she had gender, she had smoking status and various health issues. And in the analysis, she first controlled for everything to see what would have an impact, and then took out those that didn't have an impact, so that we increased our degrees of freedom. And gender was one of those that wasn't having an impact in this data, although it's not huge. But just as a signal that it's not having an impact on the results that we were looking at anyway.
Do we have maybe one more question? Shirley.
It's what you do over a number of years of lecturing and things going wrong, as we all know as academics. Microphones never work when you want them to. So I suppose the question is, and it is a small sample, and I think what the results are really interesting. So a big thank you to Beth for all the work that she's done. But I suppose it does go down to now looking at not just environmental factors, but those other factors, particularly in the older age groups, where there will be on a polypharmacy, there will be the potential of smokers, etc. And I think that will be interesting to see what influences they have on the data that you're collecting.
Yeah, absolutely. So we have got a lot of the health data, as I say, we haven't analysed yet, and that's one of the next steps for Beth to do that, subject to funding. But she's hoping to look at in a bit more depth at the health data. What I'm quite keen on is, in a future study, is to sort of measure frailty, so accumulation of deficits as a measure of vulnerability, because we know that a measure of accumulation measures of frailty basically give you an idea of how vulnerable people are going to be to adverse conditions, so like carbon monoxide as well as other things as well. So measures of frailty, because age is just a proxy basically, isn't it? Because, you know, variability increases with age. So some of our older people will be quite healthy and some of them will not be very healthy. So a measure of frailty is probably a better measure rather than chronological age anyway. Yeah, thank you.
Well, thank you again, Carol, for stepping in at the last minute. That was very kind of you. Right, I've been asked by Adrian just to let you know that we're going to have a 10-minute comfort break. We have overrun ever so slightly. I hope I'll be forgiven for that. So if we can get back here at 3:45, please, that would be fantastic. And maybe one last round of applause for our speakers this afternoon. Thank you.